Assalamu alaikum. Bismillah ar-Rahman ar-Rahim. This is the first lecture of embryology. We're going to talk about the development of human embryo. Embryogenesis means the process by which the embryo forms and develops, includes division and differentiation of cells. The human development entails a growth from one cell zygote to an adult human being. It covers the first 8 weeks of development, and at the beginning of the 9th week, the embryo is termed the fetus. The female reproductive cycle involves activities of the hypothalamus, ovaries, uterus, uterine tubes, vagina, mammary glands. The hypothalamus acts on cells of the anterior lobe of the pituitary gland, which in turn secretes the follicular stimulating hormone and luteinizing hormone. These two hormones are responsible for stimulation and control of the cyclic changes in the ovaries. The functions of the female reproductive system include formation of eggs, reception of sperms during copulation, providing conducive environment for the fertilization, and provides also shelter and nourishment to the growing embryo. First week of development includes ovulation, starts by ovulation till implantation. The ovulation means when the ovum or the eggs are released into the peritoneal cavity. It travels down the fallopian tube in which fertilization occurs. At conception, the maternal and paternal genetic material join to form the zygote. Cell division occurs with it travel down the tube and into the uterus. The ovarian cycle has two distinct phases, follicular phase and luteal phase. Follicular stimulating hormone and luteinizing hormone, they produce cyclic changes in the ovaries. They cause development of the follicles, ovulation, and formation of corpus luteum. Usually ovulation occurs around the mid-cycle about 14 days in a 28-day menstrual cycle. The ovarian follicle undergoes sudden growth and spurts under the influence of these hormones, which are the follicular stimulating hormone and luteinizing hormone. It produces a swelling or a bulge on the ovarian surface. An avascular spot appears on this swelling, which is named the stigma. The expulsion of the oocyte results from raised intrafollicular pressure. The expelled secondary oocyte is surrounded by zona pellucida and layers of follicular cells. These layers are named the corona radiata. Zona pellucida is composed of glycoproteins. This slide shows the ovary. Here are the primary follicles. There are numerous. Only one will grow and reaches maturity. This is the primary, secondary, and tertiary follicles. Here, only one will reach maturity, the other will be degenerated. As you can see, this is the oocyte inside the mature follicle, which is named the Graafian follicle. Later on, this follicle will rupture and expel this oocyte. This will be surrounded by the zona pellucida, which is a glycoprotein shell, and this is the corona radiata, composed of the cellular cells which are derived from the follicle itself. The corpus luteum means after ovulation, when the walls of the ovarian follicle collapse and are thrown into folds. This is called the corpus luteum, which serves as a temporary endocrine gland by releasing progesterone and estrogen hormones. Now, phases of fertilization. There are 3 phases of fertilization. The first one is named the penetration of the corona radiata. About 200 to 300 million spermatozoa deposited in the genital tract. Only 300 to 500 they reach fertilization site. Only one of these sperms will be able to fertilize the egg. Phase 2 includes the penetration of the zona pellucida, which is a glycoprotein shell surrounding the egg that facilitates and maintains the sperm binding to the oocyte. This induces the acrosome reaction by releasing acrosomal enzymes as acrosine. This will allow the sperm to penetrate this glycoprotein shell, thereby coming in contact with the plasma membrane of the oocyte. Releasing of lysosomal enzymes from the cortical granules lining the plasma membrane of the oocyte occurs at this stage. These enzymes will alter the properties of the zona pellucida. This is named the zona reaction. During phase 3, fusion of oocyte and sperm cell membrane will occur. After adhesion, the plasma membrane of the sperm and egg will fuse. This is actually accomplished between the oocyte membrane and the membrane that covers the posterior region of the sperm head. As you can see in this slide, this is the sperm is coming closer to this oocyte, which is surrounded by the corona radiata and the glycoprotein shell. Here it comes in contact during acrosome reaction. Here penetration occurred through the corona radiata, and these are the granules that secrete the lysosomal enzymes. Here fusion happened between the 2 plasma membrane of the head of the sperm and the oocyte. Now this slide shows the stages of fertilization. This is first stage. Usually occurs within the uterine tube 12 to 24 hours after ovulation. Here the follicle has released the ova. This is the ova surrounded by the zona pellucida. It takes it through the fallopian tube. Here the conception occurs and formation of the zygote. Then the zygote, which starts division immediately, and from 2 cells, 4 cells, 8 cells, until it reaches the 16-cell stage, which is named the morula. The division of the zygote is named the cleavage. It's completed about 30 hours after fertilization. Here the morula continues its way until it reaches the uterus. Here the blastocyst is formed due to the division of the zygote, and this will be implanted to the endometrium of the uterus. Here implantation occurs about the end of the first week of fertilization. This slide shows the fertilization again through the stages, first, second, and third. Here the fusion occurred. Here this is the last stage of fertilization. They are fused together through their plasma membranes. Here is named the 2-cell stage. Mitotic division maintains 2 diploid complement and cells become smaller. Blastomeres are equivalent. Each one of these cells is known as the blastomere. They are decreased in size. There is no growth in size of this structure here. As you can see, the zona pellucida surrounding the 2-cell stage of the zygote. Here is the 4-stage cell, fourth cell stage of the cleavage. 4 cells are formed here. The size is the same, still the same. Each one is named as the blastomere. Also here we have the zona pellucida, still the same. This is 8th cell stage cleavage. The blastomeres are equivalent in size. The results of fertilization include restoration of diploid number of chromosomes, because half of these chromosomes are from the mother and half from the father. Then when the zygote develops, the number of chromosomes returns to diploid number. The sex of the embryo will be determined at this stage, whether male or female. Then initiation of the cleavage starts here. Okay, now the zygote gives the blastomeres during the 8-cell stage. Then the 16th-cell stage, which is named the morula. Here this zygote is named the blastocyst. It's divided into inner cell mass and outer cell mass. The inner cell mass is named the embryoblast, responsible for the formation of two parts, which are the epiblast and hypoblast. The outer cell mass is named the trophoblast, which later gives the cytotrophoblast and syncytiotrophoblast. In general, cleavage means enlargement of the total cell number with a simultaneous reduction of their size, resulting from abbreviated mitotic cycle. As you can see, this is the 2-cell stage. Here they become 4, and now 8-cell stage, which is named the morula. Morula when it reaches about 16 cells in number. Here, this blastula is divided into inner cell mass and outer cell mass by accumulation of this fluid. This fluid will push some of the cells to one pole, and this will give rise to this structure. So we have now inner cell mass and outer cell mass, and this is named the blastocele. Again, here is the 8-cell stage of the cleavage. This will lead to 16-cell compacted morula, and this is scan electron microscope view of this process. Cells, then they are forced to the outside of the morula and to become the trophoblast or the outer cell mass. The inner cell mass will form the embryo proper, which later on give the embryo. This is a blastocyst. Here, the fluid starts to accumulate, and the cells they form two parts: inner cell mass and outer cell mass. This is again the inner cell mass or the embryoblast, and the outer cell mass, which is the trophoblast, is still the zonoplast surrounding this structure. And this structure again is named the blastocele, composed of inner, outer cell masses and the zona pellucida. Then, during the 2nd week, the implanted embryo becomes more deeply embedded in the metrium, and there is also further development of the trophoblast into the placenta. Also, development of the bilaminar embryo, amniotic cavity, and yolk sac. The inner cell mass will be divided into epiblast and hypoblast. Both of them represent the embryo. Now this slide shows the embryo is implanted inside the metrium of the mother. Here are the lacunae, which will be filled with maternal blood, and the syncytiotrophoblast. This is a cytotrophoblast. Here the bilaminar embryo. Here is the epiblast overlaid by the amniotic cavity, and this is the hypoblast. As you can see here, the blood fills these lacunae, the trophoblastic lacunae, and this is the embryo again, the high epiblast and the hypoblast. It will be present the bilaminar germ disc. Occurs about 12 days. Now, this slide shows that the trophoblastic lacunae and the uteroplacental circulation has begun. This is the embryo, again composed of the epiblast, hypoblast. This is the amniotic cavity, underlined also by the yolk sac. Thank you for your listening.